So how you liking this genetics so far? I hope you guys are actually really enjoying it. This was one portion of biology that kind of got me really hooked on enjoying it and really finding a lot of interesting facts about it. So I hope that you guys are kind of grasping on to some of the genetics. But I have to say, in the next couple sections, especially the last section, you'll really find a lot of information that can relate to you, and you'll be like, wow, all right, this is kind of cool. So today, section 4, so I actually combined section 2 and 3 in one recording, okay? So if you guys noticed that, I kind of changed that around. I didn't really mean to do that. I just kind of kept talking, and I realized that I went over section 2 and 3. So we're going to talk about section 4. All right, so moving on. This time we're going to take a much closer look at, what do you think? Yeah, DNA. DNA is super cool because obviously it makes who we are very unique. We're all very different, and it's all because of these really cool strands that we're going to talk about in depth today, okay? So, genetic material of cells, I want you guys to know, and we've talked about DNA briefly, we've talked about genes, we've talked about that genetic material, but we're going to go into it a little bit more depth. So, genetic material of cells, really we call genes. They're a unit of genetic material that codes for a specific trait, okay? Every single gene codes for something about who you are and what you look like. But not just physically, internally as well. So it's really kind of cool when you think of it that way. They're also, genetic material is also called nucleic acids. And you're going to see that, remember, if you remember, DNA stands for deoxyribonucleic acid, okay? And those are all the different components of DNA. Obviously, that's why it's called DNA, deoxyribonucleic acid. So we will talk about kind of each of those components as we go through with this section. DNA is made up of repeating molecules called nucleotides. Very important, because on these nucleotides is where you find that genetic material and those genes that make up who we are, okay? So of course I got to go into a little bit of history, you know. I'm not a history teacher, never was good at history, but now that I'm older, I really like it. So it's really cool to find out history of some of the science stuff. So the discovery of the DNA double helix, okay? First of all, we have a guy named Frederick Griffith, okay? He discovers that a factor in disease bacteria can transform harmless bacteria into deadly bacteria. Okay, so that's a big start because you're like, oh, all right, so this disease that can transform basically harmless things into very deadly things, there's one factor. Why would that happen? So all of these things kind of lead up to more and more discoveries. So you have to start off with something that really we look at that and we're like, so whatever. We look at what we know now, but we have to start somewhere. Then we have Rosalind Franklin. She was the first to x-ray a photo of DNA. So, hello, we got to see it. We got to know it exists before we can actually study it. So that's a pretty big thing. Good job for you, Chicky Poo. All right, and then Watson and Crick, which you guys have probably heard much about. Okay, Watson and Crick described the DNA molecule from Franklin's x-ray, which is a big deal because just a year later, and that's not too long ago, look at the years. Back here, Frederick Griffith, this is 1928. But Rosalind Franklin, 1952, and a year later, Watson and Crick are able to actually describe what she had x-rayed. I mean, that's a big deal. There's a lot to describe there. So, kind of cool, right? All right, so let's look at some visuals. There's a video that I'm going to show you that is a little bit about the history. All right, watch this.
Year 1960.
Don't even tell me you're buffering. Don't stop on me. It might. I've had these issues the last couple days. Come on, come on. I might have to post this as usual underneath if it doesn't do this. It's funny, I just watched all these videos to make sure they're working well. But, you know, that's technology for you. You got to deal with that. Okay, so I'm not going to waste time. I will just post that underneath the recording for this lesson, this mini lesson, okay? Kind of frustrating, but you know it. Same with my videos that I tried to record. Well, you guys don't know about that. Okay, shut up, Howie, right? Shut up. Okay, so let's move on. So we didn't see some visuals, but you guys will. Also, there's an assignment that you have, watch this, do this, that talks about Watson and Crick and kind of their discoveries of the DNA. let's go into the structure. Okay, so this is the structure. And remember, deoxyribonucleic acid, you have deoxyribose, which is a sugar. Okay, so you have a sugar molecule. You have a phosphate group. Okay, and then you have a sugar phosphate backbone, which makes up kind of this double helix. So down here it says DNA is a long molecule made up of units called nucleotides. Okay, this entire unit right here, see this little box here, this is a nucleotide. So you have the deoxyribose, the sugar group, you have the phosphate, and then you have this part, which we're going to go into in depth. Okay, so these are this entire kind of structure right here that makes up just 1/2 of the rung. And I mean, I call it a rung because you're going to notice that it looks like a ladder that's been kind of twisted. Okay, so 1/2 of the rung is a nucleotide. Very important. Okay, so when we talk about it a little bit more, each nucleotide is made up of 3 parts, just like I had talked about. A 5-carbon sugar called a deoxyribose, a phosphate group, and a nitrogenous base. Nitrogenous. Hello, I can never say that. So that's a nitrogen-containing base. So that's what makes up these nucleotides. Okay, that base group is so important because for the different, and we'll talk about what these little letters are that you're seeing. We'll talk about that in a moment. But each of those codes for something very different. So again, if there's any kind of a mistake in that, that can cause for some pretty big things that happen. But ultimately, that also codes for just everything about who we are. Okay. The backbone of the DNA is formed by a sugar and a phosphate group. Okay. So if you notice right here, it's showing this kind of ribbon-looking part. That's the backbone. Okay. And you can see that you have the sugar group and you have the phosphate group. Those make up the, ends of the ladder. Not the rungs, but the ends. Okay? So, the backbone. Easy, right? The nitrogenous bases stick out from the sides and can be joined together in any order, meaning that any sequence of bases is possible. Okay? So there is, obviously, certain ones go with other ones, and we'll talk about that. But that's your basic structure of DNA. Okay? So again, you always hear the double helix. Well, it is a double helix, and it's like this ladder that's kind of turned on its side. So the double helix, here are the basics. You've got this ladder, okay? Here are the nitrogen bases here, and the legs of the ladder are your phosphate and your sugar backbone, okay? But then all of a sudden you twist it, and that's not really what it looks like that. But 3-dimensionally, you have the double helix, okay? So this is if you were to untwist it, you'd have a ladder-looking kind of thing, right? Yeah. How he likes to simplify things. Okay, so here's another way to look at it, kind of the molecular structure. So a DNA nucleotide, remember you have the phosphate group and the sugar group, which basically makes up the sides of the ladder, okay, those legs of the ladder. And then you have the nitrogen bases that make up the rungs, okay? They can be, here are the different bases, and you'll hear this repeated often. There's different bases, 4 different bases when we talk about DNA. A, G, C, or T. And if you go back, and obviously you'll see these again and again, you'll notice that there are the names of them right here. Thymine is T, adenine, A, cytosine, C, G is guanine, okay? So go back down. Again, your nitrogen bases, A, G, C, T. Adenine, guanine, cytosine, thymine. Okay? You can have any combination of that. And I'll tell you about the base pairings. I keep like the suspense is killing you, right? We're going to talk more about that. But I want to give you guys a big picture of what the actual structure looks like in many different forms. So here are the nitrogen bases that we're talking about. A, T, C, and G. There are 4 kinds of nitrogenous bases, okay? They are divided into 2 classes. You have the purines and the pyrimidines, okay? So the adenine and guanine belong to the purines. The pyrimidines are the cytosine and the thymine, okay? So these are just 2 groups of bases that are going to kind of come together as the rungs, okay? And they're paired up very specifically. If they don't pair up as they are supposed to, then you have some major genetic disorders, okay? So here are the actual pairing of them. Adenine is always going to go with thymine. Cytosine is always going to go with guanine, okay? This is in DNA. Now when we get to RNA, it might look a little different, so just be aware. But anytime you have DNA and you have this strand of DNA, you're always going to have these base pairs, and that's called Chargaff's rule, okay? That's just always how it's going to be. That's how we work, okay? So the bases form the rungs on the DNA ladder by complementary pairing, okay? So adenine with thymine, A and T, always go together. Now it can be T and A, you know, it can be the opposite as well. Like you could start with T over here and A over here. As long as you have these two paired together, doesn't matter which one goes where. Then cytosine and guanine always are going to go together. So again, look at here, T and A, G and C, A and T, T and A. I can make a little song about it. Okay, just kidding. I'll be quiet. But those are the pairings. You have to know those. You have to memorize those. A goes with T, C goes with G. Okay? Do not forget those. You will see that for sure in your test. Okay? So let's go back to a little bit to Watson and Crick and what they proposed. So DNA had specific pairing between the nitrogen bases. Remember, adenine goes with thymine, cytosine goes with guanine. DNA was made of two long strands of nucleotides arranged in a specific way called the complementary rule. Okay? So they complemented each other. They always paired up as you see, just like this. So say you have this rung right here, and it's kind of small, so sorry if this is hard to read, but you have T, C, A, C, T, T, G, G, T, C. You have this complementary strand that is going to match up those base pairs, okay? So then you have A, G, T, sorry I can barely read it, A, G, T, C, or T, G, A, C, C, A, G, okay? So they all line up according to adenine and thymine, cytosine and guanine, okay? Or if you flop them, same thing, okay? So that's kind of one of the big things that Watson and Crick proposed is that there's complementary strands. They match up, okay? They're going to be that rung, those rungs, and they're going to always be in those pairs, okay? So let's talk about some amazing facts of DNA. So those, I just wanted you guys, I mean, I really kind of went through that fast, but at the same time, you guys are going to understand that DNA is not a super complex molecule. It is what it is. Everything I just showed you, it never changes. That's the way it goes. It's made up of that, but we'll talk about the replication in just a moment. So, amazing DNA facts. Woohoo! So DNA from a single human cell extends in a single thread for almost 2 meters long. That's crazy. DNA from a single human cell. Hello. Take it out of a nucleus and you just extend the single thread for 2 meters long. Okay, think of a meter stick. 2 of those. It's pretty stinking long. If you could wrap all of the DNA you have in your cells, you could reach the moon 6,000 times. Pretty crazy. DNA contains information equal to some 600,000 printed pages of 500 words each. So that's about a library of 1,000 books. That's how much information your DNA contains. That's a lot of information as I slam my table on my desk. Okay? Crazy amounts of information that you are carrying in the nucleus of your cells. Very cool. Very amazing stuff. I know that like I'm getting a little too passionate here, and it's not just because I love science. I think we are amazing creations. So, very cool. Okay. How about a tune? So this is one of my favorite songs. It's really catchy. And also I have to tell you, I had a student last year who, for her final, because I always allow people to kind of be creative. If you're not a good test taker, come up with another way to show me that you have understood all of the, not the final, she did it for the unit exam, but tell me how you understand this stuff. But she wrote a song, and I'll have to ask her permission if I can share it with you guys, because she actually made her own song on this. But this one is amazing as well as hers. I have to say, hers was kind of right along as pretty cool as this one. Okay, watch this. such a good song. As you can see, I'm jamming to it. Okay, here's another link because I don't know if it'll come back. You guys get the idea though. It's pretty. First of all, it's a really cool song. But talking about, if you guys read the captions below, and I want you guys to watch the whole thing through. So make sure you click on that and watch that. That are talking about all the effects of DNA. It's pretty cool and pretty amazing. You know, and what little things could do to alter those. Okay. So, anyways, sorry. Obviously, I'm not having the best of luck with my videos. That's life. Anyways, okay, moving on. So, how about DNA replication? Replication equals duplication. So, I want you guys to know that those are the same things. So, replication, you're like, gosh, it's such a crazy word and it sounds so intense and I don't know how I'm going to do it. How am I going to learn this? No, it's the same as duplicating DNA. Remember, we talked about it in cell division, mitosis. It happens in the S phase of interphase. So we're not necessarily in mitosis when the DNA is replicating. It's creating a identical copy of itself. Remember in those chromosomes? Remember? I know that wasn't that long ago. But that is what's happening in the S phase, is this replication, okay? Duplicating those DNA strands. Okay, must replicate before mitosis or meiosis 1. That's when it happens, always in the S phase of the interphase. That probably was on in your test, if you recall. Okay, so let's keep going and talk about that. Part of the double helix is actually unwound. Notice how basically follow these blue strands here, okay? So that was actually this one double helix, and then it's like this zipper, okay? You have, and you have two little strands here, okay? That's what we're showing right here. Okay, you have the two complementary strands that have been pulled apart, okay? So that's the first thing that happens in DNA replication. The second thing is you have these little tiny little fragments called Okazaki fragments that basically replicate parts of the strand at a time. It's not like everything is replicated all at once. It's like these little fragments come in and replicate the opposite base pairing, okay? So basically if you unzip this DNA, so you have A, G, G, blah, 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 blah on one side, these Okazaki fragments are going to come in and give that one side the base pairs that it needs, okay? And then the same thing on this other side. So it's going to give the base pairs that should have been over here. So basically it's just making two identical copies of that DNA strand, but it does it in little fragments. Like it does it and then not like this. That makes sense, okay? There's little fragments that are replicated at a time, and then ultimately you have that entire DNA strand that's been replicated, okay? So hopefully you guys understand that. We'll probably go over that a little bit more in the live class to make sure you guys are getting it. But also the enzyme then stitches those pieces together, enzymes in general. Enzymes, remember, most of you guys have already done that enzyme lab, and enzymes are so important in our bodies. They do so many different things. They are catalysts, so they make things typically go faster, but in general they just do specific functions, such as this one right here, where they actually stitch those Okazaki fragments together and basically make that complementary strand during replication, okay? So those enzymes are incredibly important because otherwise we wouldn't be able to do that replication like it's needed, okay? Remember, mitosis is occurring all the time, like constantly, at a very quick pace. So think about this. Replication is happening constantly, all right? All right, so I want you guys to think about this. In your lesson, you're going to notice that there are some different kind of theories. I want you guys to tell me, maybe you haven't read already, but which sounds the most likely how DNA would possibly replicate? Is it A, conservative replication, where somehow they produce one entirely new DNA strand? Somehow, right? Somehow, okay? How about semi-conservative replication, where this would produce two DNA molecules, each would contain 1/2 of the parental DNA along with a brand new complementary strand. In other words, the new DNA would consist of one new and one old strand of DNA. The existing strands would serve as complementary templates for the new strands. Sound pretty good? Okay, third one. Dispersive replication would involve the breaking of each parental strand during replication. Somehow The cell would reassemble the molecular fragments to create a mix of old and new fragments on each strand. Now, considering I did kind of briefly go over replication already, I'm hoping you guys realize that semi-conservative replication is the theory that is true, okay? There were a lot of these things, kind of these ideas floating around like, I think this is happening. No, I think this is happening. And what is really happening is what I just explained in this semi-conservative replication. So this nice long letter B answer, if you got that right, you're on the right track, okay? So 1/2 of the new molecule DNA is old. That's the template strand, okay? So remember, those strands, basically, they unzip, and you have basically this opening, and you have this old template, and then 1/2 of a new molecule of DNA is the complementary strand, okay? So then, again, you have these that unwind, and then you have this original strand, which is the blue, and then the new complementary strand, which is the red, okay? So, hopefully that makes a little bit of sense. Again, go and read this in your lesson if you haven't already. It should solidify when you start to see more pictures, because I try to use different images so that you guys get a bunch of different pictures and you guys can kind of start to make sense of all this information. Alright, so I have a couple of animations that I want to show you that will kind of help bring it a little bit together, because again, it's nice to see actual things. Some of you guys are real hands-on visual learners, okay? You can pretend you're like touching this DNA stuff, but at least you can see it, right? So let's watch. I've got 2 of them, and we'll see if this helps a little bit.
DNA replication. Yeah.
Sorry, I forgot how annoying this video is, but there's a point. It's good.
This is a strand of
Hang
parental
tight.
DNA. Das cytosine. Helicases travel to the origin of replication to unwind the DNA, forming a replication fork. Single strand binding proteins hold the 2 template strands apart. Next, DNA polymerase III adds DNA nucleotides to the RNA primer, but only in the 5' to 3' direction. On the leading strand, nucleotides can be added continuously. The process is different from the lagging strand. First, on the lagging strand, primase removes the single strand binding protein, via Dispersive the RNA primer. DNA polymerase III adds nucleotides to the 3' of the RNA primer. These short segments are called Okazaki fragments. Okazaki.
You're almost there. Make it through. I promise. This is still good visuals. I forgot about this video. It's okay. Whew. Now I forgot that there was a reason why I showed it is because I wanted to at least let you guys see with some commentary, which I could have done without the voices, but I wanted you guys to see, and believe me, it was kind of hard to find any good videos on DNA replication, but that gave a good kind of some images that you could see. Now, you guys will find out in just a bit what this U means, okay? So let's look at the last animation that I have for you guys. This one's a really short one, and by the way, there's a lot of details in that last video. Replication is a fairly in-depth process. Now, obviously, the gist of it is you're making this complementary strand and you're replicating, meaning you're duplicating that DNA. But there are a lot of different steps to it. So I do want you guys to know some of those details, but obviously go to your lesson and see what else is in there. But I will make sure you guys know what you need to know as far as details.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called Helicase. Spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Dun, 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 dun. Maybe I should have just shown that one, but I like that one. Little less little less details in that one, but it's kind of good to see it that way. Okay, we're nearing the end. Here's our quiz. I want you guys to think about these things, okay? Why is replication necessary? When does a replication occur, which I talked about? Describe how a replication works. And use the complementary rule to create a complementary strand on here, okay? So I want you guys to be thinking about those. I'm going to show you the answers in about 10 seconds because I want you to actually think, why is it necessary? When does it occur? Describe how replication works. Use the complementary rule to create this strand here. If you need to pause it, write down your answers and check them. So make sure you know what pairs with what, okay? So here are your answers. Remember, if you haven't written them down, pause it, pause it, pause it, pause it. Okay, here are your answers. So why is replication necessary? Both cells, new cells, will have the correct DNA. Absolutely necessary for that. When does it occur? The S phase of interphase. Describe how replication works. Now this is the quick Howie version, okay? Enzymes unzip DNA. Complementary nucleotides join each original strand, okay? And then use this complementary rule to create complementary strands. So A goes with T, adenine and thymine. G goes with C, guanine and cytosine, okay? So here's your complementary strand. All right? Okay, so there's a little bit more on DNA. How about a lot more on DNA? If you guys have any questions on any of these, please write them down and come to the live session and let me know so I can explain things further. All right?